Continue here with the Krebs cycle and aerobic respiration. So once we have the acetyl-CoA, so remember we had a preparatory step where we took the pyruvic acid and we converted it into acetyl-CoA right here. And now in the Krebs cycle, we're going to take the acetyl-CoA and we're completely oxidizing it and removing all remaining hydrogens, and then we park them onto NADH, FADH2. Also, all hydrogens will be now parked onto those coenzymes, and then what's left of the original glucose is nothing but CO2, so completely oxidized glucose. Okay, now, so here are the steps in particular. Starting out with the pyruvic acid, 3 carbons. We're removing 1 carbon. That will leave as CO2 right here. We're gaining an NADH, so this hydrogen right here. And then we end up with something called acetyl-CoA. And acetyl-CoA, as a 2-carbon compound, it will actually bind to oxaloacetic acid, this 4-carbon compound. So you can count the carbons, 1, 2, 3, 4, plus these 2 carbons from the acetyl-CoA will make our first intermediate in the Krebs cycle, and that is called citric acid. So some people also call the Krebs cycle citric acid cycle because that's the first intermediate. So again, oxaloacetic acid, 4 carbons, plus the 2 carbons from the acetyl-CoA makes 6-carbon citric acid, which then gets step by step oxidized. We will be removing 1 carbon here when we go from isocitric acid to ketoglutaric acid. So there will be 1 carbon is leaving right here, and then there's another carbon leaving right here. So remember, we put 2 carbons into the Krebs cycle, and here they go off as CO2 completely oxidized. And we're back to 4 carbon compounds that then ultimately return to be oxaloacetic acid, and would grab onto the next acetyl-CoA for making more citric acid. So the whole thing is truly a cycle that keeps going. Besides the CO2 waste product from the completely oxidized organic molecule that we originally had, we're also gaining these very important NADHs, NADH, FADH2, NADH right here. So the thing is, while you don't seem to think that we have gained anything, well, we have gained a lot. We have completely oxidized the original glucose with 2 rounds of the Krebs cycle because we are getting 2 acetyl-CoA, of course. And I apologize for the background noise here. I'm sorry about that. But here, the important part is that we have gained all of those NADHs and FADH2s. So all hydrogens have been ripped off from the original glucose. Remember, glucose, the chemical formula is C6H12O6. That means H12, 12 hydrogens, and they are now all on these hydrogen taxis, these NADHs, FADH2s. You can consider them like they're called electron carriers, but really they are hydrogen taxis or Ubers, whichever way you want to think about them. And now we're ready to move these hydrogens to the electron transport chain and harvest our ATP. So the electron transport system, it's occurring in the plasma membranes of prokaryotes and the inner mitochondrial membrane of eukaryotes. And we have these a system of electron transport molecules. So the most important ones would be all the cytochromes. Then we have flavoproteins as well, and some others. And they basically receiving the electrons. So the hydrogen will be split into protons and electrons, and then the electrons have a lot of energy, and they will be passed on in this electron transport chain from enzyme to enzyme and powering these hydrogen pumps. Basically, in the end, we are producing a lot of ATP in a process known as chemiosmosis, and we'll take a closer look at that. So here we're back at our picture we had earlier on the electron transport chain. So these NADHs, these hydrogen taxis or Ubers, they deliver the hydrogen to the electron transport chain. So the hydrogen will be split into proton and electron. The electrons will be passed on. Its flow of electrons will power all of these enzymes. And for the most part, these enzymes are hydrogen pumps. And they will pump the protons, these hydrogen ions, that was the leftover part from these hydrogens. So the electrons will be used for the electron transport chain, and the hydrogen ions, the H+, the protons, will be moved into like the space between two membranes, and if you have a mitochondrium available. So at the end, once these electrons have released all of their energy, then we will use chemiosmosis, this hydrogen ion concentration gradient that we have built up. We are going to allow these hydrogen ions to go down their concentration gradient, and that process that releases this potential energy from the concentration gradient. Once that has happened, then these hydrogen ions don't have any more energy either. And so now the protons, these hydrogen ions, plus the electrons need to be going somewhere. And for that, we need oxygen as the final electron acceptor, and we're going to end up making metabolic water. So here's the write-up to this. In chemiosmosis, you're taking the electrons from the hydrogen here, and they go down the electron transport chain, while the protons, so the hydrogen ions, are being pumped across the membrane, and that produces a concentration gradient, which represents a form of potential energy. And then once the protons have all been pumped on one side of a membrane, then we can use this concentration gradient in a process known as chemiosmosis. And I admit it's a little abstract, but just, I don't know, just try to follow through with this here. And we're gonna let them go down their concentration gradient. And through that process, this release of energy helps us to convert ADP into ATP, and we're going to make lots of it. And the molecule to which these hydrogen ions can pass is called the ATP synthase molecule. So here's what this looks like. So you have a membrane right here. You have lots and lots of these cytochromes and other enzymes that are essentially pumps. So we're using the flow of electrons, just like you would plug in your appliances, and once the electrons flow, then you can run your appliances. Here, think of these enzymes here as appliances, and they're being powered by the flow of electrons, and they're pumping. It's like a hydrogen vacuum or something, a pump in this case. And so they will take the hydrogen ions. So the NADH here delivers the hydrogen. We take the electron from the hydrogen and run the pumps. And then the hydrogen ion, the proton right here, that one is being pumped then across with the power of these pumps. So now all the hydrogen ions are going to accumulate over here. And eventually we're going to have a very high concentration of hydrogen ions that then can be utilized to make lots of ATP. And think of it this way: you have tons and tons of hydrogen ions here, and you have a little pore that's perfect for the flux of hydrogen ions. So think of it like a hydrogen waterfall. Kind of picture a real waterfall where you have the water up on the hill has a lot of energy, and you can use the flow of the water down the waterfall. You can use it to power turbines. Here we're allowing the hydrogen ions to go down this ATP synthase molecule, and as it is releasing the energy from this downhill flux, basically, we are making a lot of ATP by chemiosmosis. And you'll be surprised, it's a lot of energy. Now, once the electrons have arrived here, they have basically no more energy left. They're sort of worn out here at the end. No more energy, very low energy electrons. Then the hydrogen ions also have released their energy from this downhill gradient flux. And now we're going to put the hydrogen ions plus the electrons back together, slap it onto oxygen, and we're going to make metabolic water right here. So here, sort of the final steps a little bit more up close. You can see here the hydrogen ions are being pumped into the space right here across the membrane. So these electrons here, they're powering this pump, and they're pumping hydrogen ions into this space right here. And then once they accumulate in high concentration, then they're being allowed to go down their concentration gradient through the ATP synthase molecule. But as they're going down and releasing this energy from the concentration gradient, we're powering here the synthesis of ATP. Therefore, this molecule is called ATP synthase, and the result is lots and lots of ATP. Now, once the electrons have released all of their energy and the hydrogen ions went down their concentration gradient, and now everything has really low energy, but it's got to go somewhere. And so we're going to use oxygen as our final electron acceptor. So that means we're using oxygen to accept these low energy electrons, these low energy hydrogen ions, and we're going to put it all back together to make metabolic water, H2O. Here's the overall reaction of aerobic metabolism or aerobic respiration. So we're starting out with glucose, which has 12 hydrogens, so C6H12O6. We need 6 oxygen, 38 ADPs, and then 38 inorganic phosphates. The result is that we have 6 CO2, so all 6 carbons here from the glucose, they are now completely oxidized, no more hydrogen, so all the hydrogen is gone, it's just carbon and oxygen. So CO2, 6 CO2 plus 6 water, and we're going to make 38 ATP. That's ideal to make 38 ATP. This number varies a little bit, but we do get 2 fixed ATP from glycolysis, that doesn't change, and 2 ATP from the Krebs cycle, so that takes care of 4. And then what comes out of the electron transport chain, it varies a little bit on the exact condition, but here Totora says that we're going to end up with 38 total. That means 34 from the electron transport chain. Well, that's under perfect conditions, but nevertheless, we'll work with that. Okay, so here sort of the summary sheet. Each NADH can be oxidized in the electron transport chain to produce 3 molecules of ATP, and then for the FADH2 you get another 2 molecules of ATP. Now that's all under perfect conditions, and of course if we don't have oxygen, then everything changes. We're going to be a lot less efficient, and we don't have oxygen available as a final electron acceptor, and overall the whole process is going to be less efficient. So here in anaerobic respiration, here are some options that cells have available. They can use the following. Here's just some choices of option of possible final electron acceptors. It could be nitrate, sulfate, carbonate, and that yields the following products. Here we have nitrate, and that would be gas, nitrogen, water. Here hydrogen sulfide and water, methane gas, and water. So these are some options for anaerobic respiration. And now sort of looking at our summary shield. Now this here would be sort of the summary in terms of ATP yield for prokaryotic cells, so for bacteria. Now in glycolysis, they can get max 2 ATP by substrate level phosphorylation. Then if they do have oxidative phosphorylation available, they can convert the 2 NADH you see into 6 ATP, the best case scenario. Preparatory step, where we of course take the pyruvic acid and convert it into acetyl-CoA. We do generate 2 NADHs, and they can be converted into 6 ATP by oxidative phosphorylation, best case scenario. In the Krebs cycle, of course, we're making quite a bit more NADH and FADH2. These hydrogens, they can then be converted in oxidative phosphorylation here to 18 ATP max, and another 4 ATP for these 2 FADHs. So grand total, best case scenario is going to be 38 ATP as per Totora, and that really truly is best case scenario.